I'm excited to introduce today's lecturer, Dr. Jürgen Venitz. Jürgen is currently professor in multiple departments at Virginia Commonwealth University School of Pharmacy. He is also the vice chair of the Department of Pharmaceutics and the director of the Pharmacokinetic and Pharmacodynamic Laboratory at the School of Pharmacy. In addition, he is a fellow in the Center for Study of Complex Sciences at VCU. In 1981, Jürgen received his MD and PhD from Saarland University in Germany. From 1981 to 1985, he was the Director of Clinical Research and Development at the Institute for Clinical Pharmacology in Gunster, Germany, then completed postdoctoral fellowship training at the University of Florida. In 1988, Jürgen joined the faculty of VCU School of Pharmacy. He has published extensively and presented extensively in the area of quantitative pharmacology. I'm confident you will enjoy today's lecture.
Welcome, this is Jürgen Venitz. I'm your entertainer for today as part of the Principles in Clinical Pharmacology course at NIH. I'm a clinical pharmacologist and a professor at the Virginia Commonwealth University School of Pharmacy. My topic for today is PK/PD of protein drugs, so we're going to get into the world of biotech. So if you look at the outlines and the objectives that I put together, we talk about how different those proteins are in terms of their absorption, distribution, metabolism, and excretion, how different they are from small molecule drugs that most of us are quite familiar with. We specifically focus then in on PK/PD relationships of growth stimulating factors and monoclonal antibodies that are commonly used. And then I'll wrap up today's presentation by getting into something that is very current, and that's the approval of biosimilar drugs in the United States, which are the equivalent of generic small molecule drugs. As you can see, I have a bunch of resources that are available when you download the class material. My outline is such that we talk about ADME first, then we get into PK/PD. We'll discuss something that is fairly unique to protein drugs, and that is their ability to elicit immune response. I'm going to spend a significant amount of time talking about monoclonal antibodies, then go into two examples. One example that I'm going to lecture on, the second example that I put in as an exercise that I'll let you do on your own, and I'll review the key with you, and then we get into the biosimilar drugs. All right, so let me just start off by saying that protein drugs or large molecule drugs are very different from small molecular weight drugs that are defined as less than 1,000 Daltons. For those small molecule drugs, there are a couple of things that we know about in general. That they are subject to passive diffusion across membranes for lipophilic drugs, and or they may require, if they are polar drugs, they may require active drug transports. Okay, and we pretty much know a lot about the various drug transporters. Again, depending on their lipophilicity, they can be highly plasma protein bound or tissue bound or not. Depending on their lipophilicity or polarity, they can also be subject to primary excretion by the kidney or hepatic metabolism or biliary excretion. And their drug targets, the receptors that we are, or enzymes that we are trying to inhibit or somehow affect, are extravascular. For quite a few drugs, their tissue distribution is organ flow limited, so we can draw some fairly general conclusions, for example, about liver clearance and renal clearance. And you can see none of this is true for protein drugs. So for small molecular weight drugs, the main physicochemical property in general is their lipophilicity polarity. For protein drugs, as you're going to see in a minute, it's much more molecular weight and their particular structure, especially for glycoproteins. So here you have a general PK/PD review or overview that the drug has to get absorbed into the blood. For protein drugs, that's usually by intravenous infusion or subcutaneous injection. The drug can interact with plasma proteins, and you're going to see for protein drugs that's usually not something we worry about. They can distribute into peripheral tissues, including the tissue that contains the receptors or the targets that we call the effect compartment, and can interact and cause a pharmacological response. And the drugs are eliminated by hepatic metabolism and or renal excretion. Okay? The top part here, anything that relates to how the dose turns into a concentration, we refer to as kinetics. Anything down here, how the drug bound to the receptor turns into a pharmacological response, we refer to pharmacodynamics. Now, if you look at the absorption, that's where the first major difference starts to emerge. The protein drugs have large molecular weights, much larger than the 1,000 that are used as a cutoff, which makes them basically unable to permeate membranes, GI membranes. So they would not have any GI permeability to speak of, and sometimes even more importantly, they are subject to all kinds of chemical and enzymatic degradation in the gut. So there would be a lot of first pass effect that would prevent them from ever reaching systemic levels. Therefore, they are not suitable for all administration, and as I mentioned before, they're usually given intravenously, subcutaneously, and sometimes intramuscularly, depending on the dose and the volume. Now, even that, you have to keep in mind that if you give them by any route other than the IV route, where they're being injected or infused directly into the bloodstream, you give them subcutaneously into a subcutaneous depot or IM into a muscular depot, you don't necessarily expect them to be 100% bioavailable, because what can happen locally is degradation in those tissues. For example, proteolysis in the lymphatic system. Furthermore, so we have reduction in bioavailability, even if we give them by parenteral routes. In addition to that, the uptake or the absorption from those tissue compartments for large protein drugs in particular involves the lymphatic system rather than the blood capillaries. So if you give a small molecular weight drug, subcutaneous or IM, the absorption from the site that you're setting is by capillaries, which are relatively rapidly perfused. On the other hand, if you give the same, or if you give a different drug, a protein drug, by the same route, you now have to depend on the lymphatic system to absorb the drug into the bloodstream, which is convective by nature rather than diffusion and blood flow limited. So therefore, it's much, much more slowly. Distribution, again, due to their large size, which limits their mobility across biological membranes, most protein drugs are pretty much restricted to the vascular space for the most part. That means you would expect their volume of distribution to be, if it's purely intravascular, 3 to 5 liters for a 75 kilogram person, or if they can at least get to the interstitial space, it would be around 15 liters per kilogram. So those are, relatively speaking, small volumes of distribution that you would expect for protein drugs in general. In addition to that, as I alluded to before, they are not expected to be plasma protein bound, like a lot of small molecular weight, especially lipophilic drugs do. However, they can be taken up or bind to extravascular tissues, and that's particularly true for monoclonal antibodies that target targets in the blood or in plasma. And that would increase their volume of distribution beyond the 3 liters of intravascular space. Just like for the absorption from the subcutaneous or intramuscular depot, any extravascular distribution of protein drugs occurs by convection and possibly transcellular endocytosis, which are slow processes. So you can see here the vascular space, where along with the flow of fluid, water, and electrolytes, by convection, you have a flow of protein drugs. Okay, and you can see that the clearance by the lymphatic system is much, much faster than the clearance by extravasation, which again means that their distribution, just like their absorption in general, and I'm talking about extravascular distribution, is very slow. Now, how do protein drugs get eliminated? Again, different from small molecules, which are typically eliminated by the liver and the kidney. They are basically, protein drugs that is, are basically subject to the same pathways that dietary or endogenous proteins are subject to. So they can form a myriad of metabolites. Ultimately, the terminal metabolites are the amino acids and whatever carbohydrates were part of the original protein. And those, especially the amino acids, are ultimately recycled into their endogenous pools. So they're not necessarily excreted in the bile or in the urine. The biliary excretion or renal excretion of the parent protein, the intact protein itself, is insignificant. Now, the breakdown of those protein drugs, the proteolysis, typically occurs enzymatically, but it can occur almost everywhere in the body, including the blood. So for some proteins, they have a very short half-life in blood because they're broken down in blood, which is typically not the case with very few exceptions for small molecular weight drugs. What that means is that for some of those, especially peptides, their total clearance might be so rapid that it actually exceeds cardiac output. And I've already alluded to the fact that they can be degraded in the gut, which may be relevant in terms of elimination if they are secreted in the GI tract as well. Now here we have the first role that the molecular weight plays. So proteins that are smaller than 30,000 Daltons can be filtered in the renal glomeruli. However, so they are showing up in the primary urine. However, after that, subsequently to that, they are subject to renal metabolism. So what usually shows up in urine is not the parent drug, but some of the metabolites, unless they're reabsorbed. On the other hand, if you look at the liver, the role that the liver might play, the only way that those proteins can get in the liver is via receptor-mediated endocytosis, or RME. Okay, so they have to be taken up by particular transporters. We're going to look at some in a minute. Before they can metabolize in the lysosomes by the endopeptidases, as I said before, that are responsible for breaking down endogenous or dietary proteins as well. So right here, there's a table that I took from the book chapters that I make reference to that points out the differences. So you can see the typical small molecule drugs, 500 to 1,000 Daltons. Elimination side, liver, okay. You're talking about passive diffusion, as I mentioned before. For some of the bigger ones, there may be drug transporters involved, but the main determinant, physical-chemical, is their lipophilicity and their molecular structure. Their molecular structure would tell you what transporters and what enzymes. Lipophilicity is more responsible for crossing biological membranes. The moment you go above 1,000, so let's look at small proteins and peptides. The main eliminating organ here would be the kidney, as I mentioned before, by glomerular filtration. However, there's subsequent renal degradation or metabolism. So you wouldn't see any parent drug in urine, as is the case for the small molecular weight drugs. So the molecular weight now becomes a major determinant. For the larger proteins, so here you're looking at 50,000 to 200,000, which would include monoclonal antibodies, you have this receptor-mediated endocytosis, not only in the liver, but in other organs, as we're going to see in a minute as well. And they're depending very much on the charge and the sugar entity of those glycoproteins, because some of those transporters target mannose or fucose. The even larger proteins, they actually have to be opsonized in order to be absorbed. And if they're really big, this is larger than 40,000, they need to be taken up into the cell by phagocytosis, and particle aggregation would be the major determinant on how much of this happens. So let's look at the renal metabolism. So you can see this is a glomerulus, blood coming in and then blood flowing along the tubuli. So this would be the renal proximal tubuli. So small proteins up to about 30 to 50,000 get filtered, but then they can be broken down in the primary urine itself. They can be broken down in the brush border, or they can be taken up in the tubular cells and then metabolized or broken down in the lysosomes. Either way, there wouldn't be any parent protein that comes out in urine. There's also a possibility, for some of them at least, that the proteins actually get taken up from the peritubular vasculature into the tubule cells and being broken down as well. Either way, the only thing that would show up in the terminal, in the ultimate secondary urine would be metabolites, not parent drug. And down here we have hepatic uptake, and you can see it's not just hepatic uptake. I've already alluded to that. It's also uptake into other tissues, whether we have endothelial cells, Kupffer cells, and you can see, without going into great detail, you can see they all involved RME, receptor-mediated endocytosis. And you can see those receptors, they basically target carbohydrates, okay? Mannose, fucose, and what have you. And there are various examples of drugs that are known to be subject to those uptake systems. So this is very different from a small molecular weight drug. Now, in addition to that, for some proteins, they actually get broken down. I'm going to show you two examples for that. They actually get metabolized by virtue of interacting with their target. So they interact with high affinity and low capacity to surface receptors, and then by virtue of doing that, they're exerting their effect, but they're also being taken up in the cells and then become subject to endocytosis and lysosomal degradation. This is called target-mediated drug disposition, and we're going to use monoclonal antibody as an example that follows a similar pathway. Now, speaking about immunoglobulins, as I point out here, albumin conjugates and immunoglobulin Gs, which are basically monoclonal antibodies, or monoclonal antibodies are made from derivatives of IgG. They interact with a very particular receptor called FCRN, or the neonatal FC receptor. And it's named neonatal because the first time it was discovered, it was found to be responsible for transferring IgG across the placenta from the maternal to the fetal blood. It's also present, for example, outside the placenta, which obviously is the reason why it plays a role in the proximal small intestine, and it involves transcytosis. The FCRN, as it relates to IgG drugs, is present throughout the entire body, so endothelial cells, monocytes, macrophages, dendritic cells, all kinds of immune cells, and its role is to protect the IgG, the endogenous as well as the exogenous drugs, from lysosomal degradation, and it provides a salvage pathway, and it recycles. So this is the main reason why the half-life of IgGs is as long as it is, and we're going to discuss that in more detail. For right now, let's just see what the role plays of the FCRN-mediated recycling plays for IgG and albumin. So this scheme here compares IgG and albumin relative to their daily formation. So 7.4% of the circulating amount gets formed every day, and at steady state, 7.4% gets removed. For albumin, 11% gets formed, 11% gets removed at steady state. But how do we get to this removal or catabolic route? Well, you can see that if you look at IgG, 18% would be removed every day if it were not for about 11% being recycled. So the net result is that only 7.4% get removed. On the other hand, if you look at albumin, the difference between the net removal and the actual catabolic efficiency is much less because the recycling, the FCRN mediated recycling contribution is less. So you can see that a significant portion of catabolism for IgGs is protected by recycling the IgG, which, as I said before, leads to a relatively long half-life. Now, let's look at a couple, just schematically, a couple of elimination pathways for protein drugs. Three different kinds that I want to show here. The first one is related to the presence of ADAs, or anti-drug antibodies. So this is a phenomenon that is particular to large molecules, protein drugs, where the body develops its own antibodies towards the protein. So this would be the body creating the antibody, and the antibody is then catalyzed. So you have what we call a turnover model. Now this antibody, however, has the ability to react with a protein drug and facilitate its degradation. Okay, so one elimination would be the unintended formation, typically unintended formation of ADAs that facilitate elimination of protein drugs. The second scheme here looks at the protein drug that is, again, different from a small molecular weight drug that is eliminated not only from what we call the central compartment. So this would be the plasma compartment where the drug is being given to and it's being removed from. But it can also, this is the protein drug now, can also go to tissues and being removed from those peripheral tissue compartments. Okay, so there is a two-step elimination that is different most of the time from small molecular weight drugs. And then I already used the term target-mediated drug disposition, which is very relevant for monoclonal antibodies. So this is a little more complicated. So R would be the target or the receptor that the monoclonal antibody is intended to interact with. It's being formed continuously and it's being eliminated continuously, so we are at steady state. Now this receptor then interacts, you can see, with an equilibrium reaction with the protein drug, and you now have a second elimination pathway for the receptor. So the receptor is endogenously removed, but it's also removed after interaction with the monoclonal antibody. And obviously it is that effect that is intended by giving the protein, the drug, meaning the monoclonal antibody, to hit the receptor. Now the reason why that's important, those processes can be saturable, meaning dose dependent, and they can be time dependent. We can have nonlinear PK, which is for small molecular weight drugs the exception. The example that I'm showing here, we are looking at the plasma concentration on a logarithmic scale versus time, and a macrophage colony stimulating factors, MCSF, is given by IV at 3 different concentrations. So you can see this is the low concentration, the intermediate concentration, and the high concentration. And what you see here is what we like to call in PK lingo the hockey stick, okay, which is typical for a nonlinear or saturable drug. So initially there is a distribution, there's a plateau phase, and this is when the concentrations drop below the saturation levels and we have a relatively rapid elimination. So you can see for all those 3 doses, the most rapid elimination occurs at low concentrations that are below the saturation level, and any higher concentrations lead to this plateau. Now, in addition to this propensity to be associated with nonlinear PK, at least for some of the protein drugs, their PK-PD relationship, their relationship between the concentrations in blood and the target effects can be very, shall we say, indirect, meaning there are multiple steps between achieving drug levels and actually observing the effect. So for example, as I point out here, for growth stimulating factors, which would include erythropoietin, EPO, filgrastim, the example that I used later on, and the one that we just talked about, the MGCSF, their PK/PD typically follows what I call the indirect effect model, meaning there is a pronounced lag time between plasma concentrations and pharmacological effects based on their mechanism of action. So let me just illustrate that by using erythropoietin, which, as you know, stimulates the formation of red blood cells. So what you have here in blue would be the pharmacokinetic part of this model. So erythropoietin is given into a central compartment and it's being eliminated, and this would be a saturable elimination pathway characterized by Vmax, the maximum elimination rate, and Km, which would be the saturation level or the affinity constant that I alluded to earlier on. So this is the kinetic model. Now, how does erythropoietin lead to increased red blood cell levels in blood? But it does that in the bone marrow by stimulating precursor cells. So P1 and P2 are precursor cells, or are the reticulocytes, which are the immediate precursors to red blood cells. So each of those precursor cells leads to, by maturation, leads to the next level. They all have a finite lifespan, which is what the tau relates to. Okay, so you can see there is an effect that slowly occurs because each of those stimulations has to translate into increased concentrations of the subsequent red blood cell or precursor. In addition to that, as alluded to here in the scheme, there is a negative feedback system that if the reticulocytes go up, meaning a lot of red blood cells are going to be formed down the road, that actually downregulates some of their precursors. Okay, so what that means is that to get from the drug level to the effect, there's a whole chain of events that have to occur that take time. And we have nonlinearities built in. And you can see in this particular example, the stimulation relationships, that's what the ST stands for, both follow what we call Emax relationships, so they can be saturable depending on how the drug concentration compares to the effective concentration, the EC50 value. To illustrate that, to get away from the scheme and actually look at data, on the top graph, so you're looking at multiple doses of erythropoietin in humans, and you're going to look at reticulocytes on the top, red blood cells at the bottom, and hemoglobin at the very bottom. Okay, and you can see that with each administration of erythropoietin, the reticulocytes go up and they peak after about 200 hours. And then they're being basically maintained until the last dose is given and they drop off. On the other hand, if you look at the red blood cells, nothing really changes until maybe 200 hours. So while those reticulocytes go up, we don't see the red blood cells yet because they haven't matured, or the reticulocytes haven't matured yet. Then they mature and the red blood cells continue to go up even as the reticulocytes decline. So it is the lifespan of those red blood cells and the maturation of reticulocytes into red blood cells that makes the ultimate response, which is the increased red blood cells or increase in hemoglobin, much, much slower than the primary response, which would be the increased reticulocytes. Similar to that, another growth stimulating factor, so now we're looking at filgrastim. Again, that's going to be my example drug in a few minutes. GCSF. In blue, we have the PK model. So here we have the drug being given into a one-compartment body model. The main difference to EPO, we now have two routes of elimination, what's called a linear route and a neutrophil route. So this drug is metabolized via binding to this neutrophil receptors that I'm going to discuss later on. This is a sessionable process. The linear clearance is elimination by the kidney because it's a relatively small molecule. Now, how does that relate into pharmacological effects? Again, we are looking in the bone marrow at precursor cells to the white blood cells or the absolute neutrophil count that we can measure in the peripheral blood. And the stimulation to the precursor cells takes time depending on, and you can see some of their lifespans, 40 hours, 60 hours, 95 hours. So that's how long it takes after you stimulate those precursor cells before they show up in plasma or in peripheral blood as neutrophil count. And here's the corresponding plots. So on the left-hand side, you're looking at the serum or plasma concentration versus time, single doses after subcutaneous injection. You can see the concentrations increase with higher doses. Their terminal slope seems to be very similar. Okay, on the other hand, if you look at the pharmacological effect, so this is the absolute neutrophil count versus time, you can see with increasing doses, the peak effect gets later and later. So at the highest dose, it takes 6 days for the neutrophil count to achieve its maximum, even though the peak plasma concentrations are achieved within 2 days. What's the reason? Well, the reason is that the effect has to work its way through the various progenitor cells. Okay, so this is an indirect effect model, and it's the cytokinetic model, meaning the time, the lifespan, and the various transfer constants between those precursor cells drive the time profile in the pharmacological response. Okay, something unique, unrelated to the PK/PD of protein drugs, is their ability, as I mentioned before, to cause immunogenicity. So they can cause immune responses, anything from anaphylactic shock to neutralizing antibodies. And I've listed here a few potential consequences. So you can have loss of efficacy. For example, if you have patients with hemophilia and they develop antibodies to factor 8, they're basically unable, even with high doses, to use factor 8 as a way to replace the missing clotting factor. Okay, and you can see some of the monoclonal antibodies, and we'll talk about that in the exercise. On the other hand, sometimes those immune responses, meaning those anti-drug antibodies, can actually mimic the drug itself. So growth hormone, the antibodies themselves also have, as a pharmacological response, similar to what growth hormones does. They can neutralize endogenous proteins. This is the story about EPO. A while ago when they changed the formulation of EPO, I think they changed the rubber of the top of the glass vial that had EPO. It changed the structure of EPO, and EPO became now immunogenic, and patients that received that erythropoietin actually developed antibody to erythropoietin that targeted not only the exogenous but also their own erythropoietin. so they were completely unable to produce red blood cells because their endogenous protein was neutralized by those ADAs. And then I already mentioned allergy, anaphylaxis, and serum sickness if you have lots of circulating ADA complexes with the protein drugs. This is just from a kinetic point of view, the formation of ADAs can actually typically increase clearance because you're now removing protein drug from the circulation, so they can actually reduce the activity. In rare cases, they can also decrease the clearance and increase the activity. But that's the exception. The main concern is that you have ADAs, that the clearance of those protein drugs is enhanced and their activity is reduced. Where does the immunogenicity come from? Well, we've already talked about potential differences in sequence, especially when we use non-human amino acid sequences, as we do for some of the monoclonal antibodies. Their glycosylation pattern, I think this is something that is currently investigated very intensely to find out what carbohydrate change in what position might induce immunogenicity, because if we understand that, we can actually avoid it. And then the other thing to keep in mind is that the proteins themselves can interact and form trimers, tetramers, what have you. Okay? And there are impurities and contaminants as a result of the biological synthesis, formulation, technology. All this goes into the potential risk to cause immunogenicity. For the purpose of this lecture, we're going to talk about the glycosylation pattern and the sequence variation a little bit later on. Okay, now let me switch to then a particular kind of protein drugs that really have had a major impact on therapeutics, and those are monoclonal antibodies. The reason being is they're so highly selective and have such a high affinity for their intended target that they're about as close to the magic bullet that Paul Ehrlich stipulated in the early part of the 20th century. So they're about as close to just hitting the target rather than having off-target effects, even though, as you're going to see in a minute, they all have potential for non-target related risks. So what's the basic structure of a monoclonal antibody? As I mentioned before, they're basically, at least the ones that are currently approved, are IgG drugs. So if you look at the molecule here, it looks like the only multimedia show for today. They look like a VCU cheerleader or like a Y. So you have the FC part of the molecule down here, and you have the FAB part of the molecule, are the two ends of the Y. The top part here, that's where the antigen binding region is, composed of a light and a heavy chain. So this is what makes the specificity of each of those monoclonals. You can see they are connected, the two chains and the various sections of the protein are connected by disulfide bonds. And as I mentioned before, the FC segment, the stem if you like, that's responsible for the effector function, whereas the FAB section is responsible for the antigen. I also point out, and it's highlighted here in those green circle, that there are lots of carbohydrates in those monoclonal antibodies. So they are large molecules and they are glycoproteins. This is to give you some idea how they compare across the various classes. So I'm only going to focus in on IgG, because as far as I'm aware, those are the only ones that are therapeutically available, but I want you to realize that endogenously we have IgA, those are typically on epithelial surfaces. IgM, those are the macroglobulins, IgD and IgE, those are related to histamine release on mast cells. But the monoclonal antibodies are typically from the IgG class. So let's see what they have in common. You can see their molecular weight, as I said before, relatively high, 150 to 160,000. You can see their half-life is to the tune of 3 to 4 weeks, with the exception of IG3. Okay? Now, what do they do in terms of their effector function? They all activate to some extent the complement pathway I'm going to talk in a minute about. They do not activate the alternative complement pathway. They are not present on mature B cells. Probably the most important thing to keep in mind is their ability to bind or not to bind to the FC receptors. Remember, those are the receptors that contribute to the recycling and the prolonged half-life. Okay, so IgG1 is the most commonly used backbone for drugs that you want to target and knock out, because they allow you to bind to the target and induce effector to remove it. Now, in terms of the convention that we use to name them, again, this is different from small molecular weight drugs that all have their pretty generic name. We have murine antibodies. They would be MOMAP. I'm not going to talk about them because they all have an unacceptable risk of causing immune responses. However, we do have chimeric antibodies. So you can see those are antibodies where the FAB, the antigen binding part of the molecule, is murine, meaning based on mouse sequence. The rest of the molecule is human. We have humanized antibodies, the ZooMaps, that have basically small sequences of murine, but they are fully humanized. And then we have the gold standards nowadays, which would be a fully human antibodies, where both the FAB and the FC sequence are all human. So you can see adalimumab, which is one of the TNF antagonists, is a human antibody. And you can see that infliximab, which is another TNF, that's a chimeric antibody. Now, what are the effector processes? In other words, once those monoclonal antibodies hit their target, how do they remove it? Either by complement-dependent cytotoxicity or by antibody-dependent cellular toxicity, CDCC or ADCC. Let's look at CDC first. So here you can see that's the monoclonal antibody, the little Y, that binds to a target. In this case, it would be a surface target. This would be a CD20, which is a target for rituximab. By binding the FC receptors, so the bottom part, if you like, the stem of the molecule, activates complement. Complement proteins gather and they form what's called a membrane attack complex, a protein complex that basically opens up a pore, and through that pore, basically, you have the extracellular and the intracellular fluid being in contact, and the cell is going to be lysed. Okay, so the binding of the monoclonal antibody to the target leads to FC-mediated complement activation, which kills the cell. That would be CDC, complement. On the other hand, if you look at the ADCC, you now have, again, a target that the monoclonal antibody has bound to, and now the FC, the bottom part of that molecule, interacts with FC gamma receptors that are present on other immune cells. In this case, would be a macrophage, a monocyte, or a natural killer cell. Okay, so here the monoclonal antibody hits the target, and then it activates other immune cells to destroy the target and the cell associated with it. So this requires complement. This requires other immune cells. The last thing that I want to review with you in more detail is this FCRN-mediated recycling. So regardless of what target IgG or monoclonal antibodies have, they are subject to FCRN or the neonatal FCR receptor recycling. So how does that work? Well, this would be the IgG, could be the endogenous IgG, or it could be the monoclonal antibody. It binds to the FCRN on the cell surface. It forms an endosome, and the endosome gets internalized. Okay, so you now have inside the endosome, you have the IgG, both free, and the IgG that is bound to the FCRN. The IgG that is free is then metabolized, broken down in the lysosome. So that's gone. On the other hand, the FCRN-bound monoclonal antibody is protected from this degradation, and it gets recycled to the cell surface where the antibody is then released again. So you have recycling of portion, and you have breakdown of some of it. And remember, this is not related to the FAB. This is not related to the antibody targeting part of the molecule. This is all related to the FC portion of it. So if you put this together, monoclonal antibodies are subject to two parallel elimination pathways. The first one that I've already alluded to is TMDD, or target-mediated drug disposition. So here the monoclonal antibody, by virtue of its FAB, forms an equilibrium with the drug target. It tags it, and then you have subsequent elimination via FC-mediated processes. So this could be CDC or ADCC. This is a pathway that is highly selective. It only occurs if the drug binds to the target. And it depends on how much target and how much drug there is. In other words, it is saturable and depends on the concentration of both of the contributors to this complex. But in addition to that, any monoclonal antibody is also subject to this relatively non-selective FCRN mediated. Okay, so here with the FC part of the molecule, you have endocytosis and subsequent degradation. However, any of the monoclonal that is bound to the FCRN is protected and gets recycled. So this is a pathway that does not depend on target concentration because the target is not involved, and it is usually not saturable. However, something that I didn't point out here that I want to point out verbally is that this process happens not only for monoclonals but also for endogenous IgG. So you have endogenous IgG, the patient's IgG, potentially at least, compete with monoclonal antibodies for those binding site, which then could prevent them from being recycled. The reason why this is important from a kinetic point of view, the combination of those pathways, along with the fact that the target itself changes over time, means that there is a potential for dose and time-dependent pharmacokinetics. So let's look at an example where we work out those various relationships. So this is an antibody that targets a surface receptor, CD11A, on T cells. Okay, so you have the free antibody that interacts with its target. It forms a complex, and this is a reversible interaction. However, this complex itself is now subject to this, as I mentioned before, TMDD, so specific saturable receptor mediated clearance. And you can see there are various FC receptors that lead to the removal of the target that is bound to the monoclonal. In addition to that, the monoclonal by itself can also be removed by this non-saturable FCRN pathways. Okay, so this is non-saturable, this is saturable, and depends on how much target there is. So if you look at, again, the concentration in plasma versus time, for the different doses from 1.2 to 10 milligrams, so this is a 100-fold range in doses, you see again what I like to call the hockey stick, that with higher doses you get a plateau, but the terminal rapid elimination is the same across doses because now the concentrations are below the saturation level. And I didn't put the numbers here, but if you look at the clearance values across those doses, you can see that from the lowest to the highest dose, the clearance is reduced 60-fold. So this is strongly nonlinear. You also see an increase in, a decrease in volume of distribution, only about 2-fold. That means with increasing doses, you have saturated now the binding sites, not only the removal sites. Okay, so this is typical for TMDD, that both clearance and volume go down. Now, if we look at the PK-PD relationship, so this is now not after IV, but after subcutaneous administration of one of those doses. And we're looking at the left-hand side at the plasma concentration, and here we are looking at the concentration of the target on the right-hand side versus time. So you can see after subcutaneous administration, the monoclonal antibody concentrations in plasma go up, they peak, and they decline. Consistent with that, you can see that the target concentrations go down as well. Okay, so the drug hits the target, that binding is almost instantaneously, and the target gets removed. And then as the monoclonal antibody concentrations decline, meaning as that monoclonal antibody complex is being removed, you have a recovery of the target. So the target comes back because the removal now gets less and less and the target gets regenerated. Okay? And you can see that is true whether you look at the expression or you're looking at the binding sites. Okay, so here you can see that the target concentrations have a direct, or the target levels, I should say, have a direct correlation with the concentration of drug. But they also depend on the turnover of the target. Underneath, you have now repeat those studies in patients with psoriasis. The idea being is that by hitting the target, by knocking out those T cells, that the psoriasis, an autoimmune disease, should be benefiting from that. So what you're looking at here is, again, over time. Now look at the different time scale because you're now looking at repeat doses once a week. On the left-hand side, we're looking at the drug concentrations. So you can see weekly doses, the drug concentrations, they go up and then be at steady state, and at the last dose, the drug concentrations decline. In yellow, we're looking now at the target, and the target is on the right-hand scale. And you can see that with the first dose, the target is knocked out, and it remains knocked out basically until the last dose is given, and then the target gets regenerated. It comes back. Okay? And you can see that the target and the free binding sites, just like in this plot here, they're basically parallel. If we now relate that to the clinical effect, that's what the clinicians care about. They have what's called a PASI score, psoriasis area and severity index, and that's in purple. And you can see that that score, even though the target gets knocked out right away, that score changes very slowly. And even after the last dose, that score only slowly returns back. So there is a further delay from translating the knockdown of the target, the CD11A, to relating that to the clinical symptom, which tells us downstream of this target there is pathophysiology that basically explains for this lag as well. Okay, let me go through one example, and the second one, as I said, is your exercise. So the first example is a growth-stimulating factor, filgrastim, GCSF. And I want to start off by showing you how different this protein is from a monoclonal. Okay, you can see it's much smaller. Okay, it's about 20,000 Dalton. Monoclonal is about 150,000. It is non-glycosylated, so we don't have to worry about various carbohydrates. It is a single chain protein, as opposed to the two light and the two heavy chains in a monoclonal. Okay, so filgrastim should be much easier to deal with in terms of the manufacturing than a monoclonal. What does it do? Well, as I mentioned before, it binds to a specific receptor on the surface of white blood cells and their precursors. And by binding to the cell surface receptor, GCSF receptor, that gets translated in cell proliferation. So it leads to increased maturation of white blood cells. However, and the reason why I use it as an example, it has a very complex kinetic properties because it is subject to non-saturated renal elimination because it's a relatively small protein. And then it has a second pathway, and that's the one that causes trouble because that pathway depends on how much receptor there is. So this binding to the receptor not only leads to the intended pharmacological response, increased white blood cells, but it also leads to the breakdown of the drug in the first place. So it's limited, this elimination is limited by how many white blood cells there are, and therefore it becomes saturable and time dependent. So let's look at a couple of plots to illustrate that. And they are part of one of the handouts that I've included with the class material. So let's look at after IV administration. So here, filgrastim is given intravenously as an infusion, I think over 30 minutes. And you can see after a single dose, it doesn't look like hockey sticks, so it looks like it might follow linear PK. If you look underneath the pharmacological response, the absolute neutrophil count versus versus time, you can see that the absolute neutrophil count goes up, peaks, and then it goes down. Main thing, as I alluded to before, the plasma concentrations peak right at the end of the infusion after 30 minutes. However, the white blood cells don't peak until after about 20 hours. So again, we have this disconnect, this lag time that is caused by the maturation, the cytokinetic model. On the right-hand side, we are now changing the administration route, and we're giving filgrastim subcutaneously, so you now can see concentrations in plasma go up, they peak, and then they go down, maybe with a longer half-life, so maybe there's a little flip-flop. Underneath, we can see, again, the absolute neutrophil count goes up and it peaks, and you can see the change here is less than it is after IV because we don't have complete bioavailability. You can see after subcutaneous administration, the peak levels are about 10. After IV, they are more than 100. More importantly, when we look at repeat dose administration, so now we're giving the drug subcutaneously repeatedly at different doses. So at the very top, we are looking at the plasma concentration of the filgrastim versus time. So this is the first dose, second dose, and so on. And let's just compare for the low dose, that's 2.5 micrograms per kilogram, the first dose and the last dose. And you can see that at the first dose, the drug levels are higher than after the last dose. Let's look at the higher dose, 5 micrograms per kilogram. The first dose has higher doses than the last dose. And the same is true for the highest dose. So over time, the drug concentrations behave very different from what you would expect. There is no accumulation. There's the opposite. There's a loss of concentrations over time. All right? Let's look underneath what happens to the pharmacological response. So here you're looking at the absolute neutrophil count versus time, and you can see the absolute neutrophil count goes up, and the highest level are achieved as you would have expected after the last dose. Okay, so you can note that at the first dose, when we have the highest drug level, we also have the lowest white blood cell count. At the last dose, when we have a reduced drug levels, we also have the highest white blood cell count. And obviously, this is the explanation. As I mentioned before, the elimination, at least part of it, the major part of it is, of filgrastim depends on how many neutrophils we have. So as we have more neutrophils, the clearance is increased and we have lowered or reduced levels. Okay, so when you look over time, so this is based on a model that they used in this particular paper. If you look at the amount of receptor that is available for the drug to bind, to exert its effect, but also to be eliminated, that receptor goes up. Or if you look at the better way to look at it, if you look at the clearance, the instantaneous clearance over time, you can see within each dose, the clearance, because it's a saturable clearance, goes down and then it recovers prior to the next dose. Okay? But in addition to that, over time, because you now have more white blood cells, you also have an increase in residual clearance. So at the last dose, the clearance is much higher than after the first dose. Now, so that's complex, time and concentration dependent. Now, one of the advantages, or one of the disadvantages of filgrastim is it has to be given fairly frequently. So attempts were made to improve that, and one way to do that is by pegylating it. So this is a covalent binding of filgrastim to polyethylene glycol that increases the molecular weight to above 30,000, which basically shuts down the renal excretion. And if you go back, we are shutting down this pathway, and the pegylated filgrastim now has only one way to leave the body, and that is via this receptor-mediated degradation. So again, let's look at a study. So this is a study on top. We're looking at the concentration of the pegylated filgrastim, and underneath we are looking at the neutrophil count. And I'm going to explain to you in a minute what happens to those neutrophils. So you can see first that the concentrations increase dose-dependently and stay around longer than after the non-pegylated. So the non-pegylated are the triangles, and you can see all those half-lives or terminal decline is more slowly. So we can give it less frequently. That's obviously the intent. Now, if you look at the ANC, we now have to appreciate that those studies were done in patients that underwent chemotherapy. So they got a test dose, if you like, or a pre-chemotherapy dose, and they saw a time-dependent increase in neutrophil count. On day 15, they received their chemotherapy, which was highly myelosuppressive, so you would expect the white blood cells to go down. A day later, they got their second dose of filgrastim. And you can see that even though there was a drop in white blood cell because of the chemotherapy, that drop was actually the less with the pegylated than it was with the non-pegylated. Okay, which again suggests that the difference in half-life, that this non-pegylated has such a short half-life that it does not cover all the post-chemotherapy myelosuppression. If you look at the clearance values, again, to illustrate how nonlinear the kinetics has become. If you look at those were the patients that I just reviewed for you with non-small cell lung cancer before chemotherapy, and let's just look at the clearance. With increasing doses, the clearance declines from 64 to 7. So this is a sign of the concentration or dose-dependent kinetics. We are saturating the pathways via the white blood cells. After chemotherapy, so this is when the chemotherapy now has reduced the white blood cells, the clearance across all doses is further reduced because we basically have reduced their elimination pathway. We still have dose dependence. And what I've done here is I've actually plotted the area under the curve. This is for the drug in plasma versus dose. And I've plotted the area under the effect curve, which is the ANC count versus dose as well. So let's look at the kinetics in blue. And you can see that this is not a straight line, but it is a super proportional relationship, meaning with increasing doses, the area increases more than proportional to dose. This is true for the kinetics. And the reason for that is that we have saturation. However, if we do the same plot for the pharmacodynamic response, you can see that the pharmacodynamic response is actually infraproportional. So increasing the dose, we get a less than proportional increase in the area of the ANC count. That has to do with the fact that those doses are already saturating the maximum possible response. So for the purposes of dose selection, as the paper that I have included with my handouts points out, they looked at the relationship based on a model between the maximum effect, okay, as a function of the concentration of filgrastim. They defined it, by an EC50 value of about 8 nanograms per ml. And you can see the various doses, the top 2 doses, basically fall into the plateau portion. And you can see those original doses in the studies that I just reviewed for you were body weight corrected, and they actually then picked a non-body weight corrected dose that allowed them across a fairly large range of body weights to fall into the plateau. All right, so filgrastim has a reduced total clearance relative to filgrastim because we're taking out the renal elimination pathway, and it depends exclusively on the neutrophil-mediated elimination pathway. As a result, the pharmacokinetics becomes strongly nonlinear and saturable after single doses. However, as far as the dynamics is concerned, we can select doses that fall into the plateau, and as a result, we get those effects that we're talking about here, preventing the chemotherapy-induced myelosuppression. The TNF blockers, we'll talk about that after the end of my lecture. So let me now wrap up my presentation by talk about biosimilars. So biosimilars are the equivalent in the biotech world of generic drugs. The idea basically is if we can classify a biosimilar to a reference protein drug, they are therapeutically substitutable. Okay, another term that you find in the literature to refer to that are follow-on biologics. Now, just to give you a reference, no pun intended, generic drug products for small molecular weight drugs basically depend on a comparison of the area under the curve and bioequivalence assessment, okay, in order to conclude that they are therapeutically substitutable unless they are locally acting drugs. However, for biosimilars, because of the complexity of the molecule itself, that's not sufficient. So the approach is different. And you can see this is the pyramid that you find lots of time that's supposed to help understand how we conclude that a biosimilar is biosimilar or highly similar to the reference product. Most of this pyramid refers to analytical tools that allow us to characterize the molecular structure, the physicochemical properties, the in vitro properties in terms of the FC and the FE part of monoclonal antibodies, for example. So this is all based on analytical work. Then there are non-clinical studies to show that the PK and, if possible, the PD in animals are comparable. What I want to focus on are the top 2 parts of the pyramid. We want to show, so in order for a biosimilar to demonstrate biosimilarity, the reference and the potential biosimilar have to show that they are bioequivalent kinetically, bioequivalent dynamically, and that they are clinically not distinguishable. So as we move up, we are reducing uncertainty. Uncertainty meaning we are convincing ourselves more and more that those two products are truly therapeutically substitutable. And as I point out here, PK/PD, including immunogenicity studies, they are intended, so that's the top part here, they are intended to show that any differences between the two products are not clinically significant between the proposed biosimilar and the reference product. And that's all part of the totality of evidence, which would be the entire pyramid. All right, now for the PK/PD part, typically we have clinical pharmacology studies in healthy volunteers. So those are not patients, but study subjects in order to demonstrate PK and PD, if possible, bioequivalence. In addition to that, this is required in the United States per guidance. In a phase 3 study with patients, we have to demonstrate, or it has to be demonstrated, that there is no clinical difference between outcomes, or for outcomes between test and reference products. And as part of those studies, there's usually a sub-study where the PK in patients will be assessed. And lastly, clinical immunogenicity should be comparable. So what I've done in the next few pages, I have summarized the 4 biosimilars that as of right now are approved in the US. And you can see one of them is a GCSF that we've already talked about, and 3 of them are monoclonals. Okay, so we have filgrastim, just talked about that. That's one biosimilar approved. And then we have 3 TNF blockers that are part of the exercise that also have been approved. And you can see how were they studied. Well, they all underwent a pharmacokinetic study in healthy volunteers, either as a crossover or parallel group. So there was washout involved for the crossover studies to make sure that the 2 treatments didn't carry over. You can see that in all, across the board, both US and EU sourced reference product was used. So those are all multinational companies. They wanted to target approval in both the EU and the United States. You can see fairly large sample sizes to demonstrate bioequivalence. On the next page, I've listed, according to the label, whether the drugs follow linear or nonlinear PK. And as we talked about for filgrastim, it is subject to a strongly superproportional PK. All the other, at least 2 of them, are known to be linear in the therapeutic range. The third one is not stated in the label. As a result, you can see that for the filgrastim biosimilar, different doses and different routes were studied to deal with the fact that there are superproportional PK. On the other hand, if you look at the monoclonals, pretty much straight single doses and a fixed dose, a therapeutic dose was studied. All those studies used bioequivalence, so the area and the Cmax ratios between biosimilar and reference had to be within 80 to 125%. In addition to that, and I think this is kind of tough to see, for all of those 4 biosimilars that are approved, phase 3 studies were done in patients. The dosing regimen that was used was obviously a therapeutic dosing regimen, and in subgroups of patients, at least informally, the pharmacokinetics was assessed using either areas under the curve during a dosing interval or trough levels. Other than the ANC count for filgrastim, for any of the TNF blockers, no PD metrics was assessed. As far as immunogenicity is concerned, you can see that of the 4 approved, the immunogenicity rates were, quote, similar meaning in the eyes of the FDA and the sponsor, not different between the biosimilar and the reference. For one of the two, or for one of the four, a post-marketing study actually was required because there were differences that could have been clinically significant. So let me walk you through an example, and I picked the Soxio, which was the first US approved biosimilar. I picked that as my example. Okay, so the biosimilar needed to demonstrate biosimilarity for the US approved reference product, Neupogen, included PK/PD studies. So let's walk through them. One of the phase 1 studies, one of the PK bioequivalence studies that I alluded to in my summary sheet, now looks at the concentrations of filgrastim versus time for the Soxio, which is now the biosimilar, and the reference product, the Neupogen. So here you can see that the profiles seem to overlap nicely. The formal bioequivalence assessment tells us that both the area under the curve and the Cmax, their 90% confidence interval, falls within 80 to 125%. So they passed the pharmacokinetic bioequivalence. In addition to that, because we have a marker, a pharmacodynamic marker, as part of this study, they also measured the pharmacodynamic bioequivalence. And in order to do that, for the same study now, they measured over time the ANC, the absolute neutrophil count versus time. Again, you can see the profiles are virtually superimposable, using the bioequivalence criteria for both the area under the effect curve and the peak effect fall within the bioequivalence goalpost. Check. Then I mentioned before, given the fact that there is a relatively complicated dose dependence, they also looked at 2.5, 5, and 10 micrograms per kilogram sub-Q. Again, you can see without any formal testing that the dose dependence for both the Soxio biosimilar and the Neupogen reference are basically superimposable. So in all the clinical, the phase 1 PK/PD bioequivalence was achieved. And then to confirm that this translates in patients in any or in no clinically significant difference between the two. So they did a study in patients with breast cancer that underwent chemotherapy, and they received 2 doses of filgrastim. So just like we talked about before, this is the first dose of filgrastim. Then somewhere here they received chemotherapy. You can see the white blood cell count declines, and then they get the second dose. And you can see again, if you compare the Soxio, the biosimilar, and the Neupogen, the reference, the profiles are virtually superimposable. And the clinically relevant outcome that was used to assess the study was the duration of severe neutropenia. So if you look at, for the both treatments, the mean and the 90% confidence interval, they basically overlap, and they could conclude that the two treatments are bioequivalent. So based on the totality of the evidence, they were able to show that Soxio and Neupogen were biosimilar, and Soxio in 2005 became the first biosimilar ever. Now that concludes my formal presentation. I'll give you a chance to work on the exercise, and if you'd like to, you can listen to me again, and I'll tell you what you should have gotten in the exercise. Hello, this is Jürgen Venitz again. I'm ready to review for you the exercise that I had asked you to do on the TNF antagonists. So let me just briefly review what I had asked you to do. So I had posted the prescribing information for those 5 TNF blockers, and I wanted you to look at different sections, section 6, 7, 11, and 12, in order to summarize the differences between the products, their molecular origin, mechanism of action, recommended dosing regimen, use in the geriatric population, notable drug-drug interactions, basic quantitative information, and immunogenicity. So what I have put together for you as a key is a summary. So let's just look at across from left to right. You can see we have adalimumab, infliximab, etanercept, golimumab, and certolizumab, PGOL as the 5 TNF blockers. So what they all have in common, they are targeting tumor necrosis factor, which is an inflammatory protein involved in autoimmune diseases. So let's see how they compare in terms of the type. And you can see two of them, adalimumab and golimumab. They are recombinant IgG1s. They're targeting TNF, and they are completely human. Umimab and umumimab. Okay, so they are human, completely human proteins. Compare that to infliximab. You can see there's an XC. So this is a chimeric protein. So here we have the IgG1 kappa chain connected, this is human then, connected to the murine variable. So there is a mouse component to this, but it's still a monoclonal antibody. Then we have etanercept. Well, etanercept is not a monoclonal antibody. It's what they call a decoy receptor. It is a fusion protein where the TNF receptor is combined with FC of IgG1. So they're taking the receptor, the human receptor that is available recombinantly, and they are bioengineering then a protein where they're combining it with the FC receptor. Why would they combine it with the FC receptor? Well, remember the FC receptor is responsible, among other things, for FCRN mediated recycling. So they're basically doing this and take advantage of the FCRN to prolong the half-life. Otherwise, the TNF receptor by itself, that protein, would be degraded very quickly. And then the last one, the certolizumab, that's a humanized, so it's not human but humanized, so it has some murine sequences, and it's conjugated with polyethylene glycol. Why did they conjugate it with polyethylene glycol? Again, to prolong the half-life. So this is a monoclonal antibody, but they're trying to prolong the half-life. So if you just switch down and look at the, let's look at the mechanism of action first. So all the monoclonals, they are inhibiting the receptor, both soluble and membrane-bound. The one exception is the decoy receptor. Okay, so that is the receptor that you're giving exogenously that is competing for TNF, and it's sucking it up and it's preventing it from doing its damage. Other than that, they're all TNF binding / neutralizing molecules. How do they compare in terms of their different pharmacokinetic properties? Well, the first thing, infliximab is the only one that is given intravenously, so we obviously don't have to worry about bioavailability. The ones that are given subcutaneously, you can see the bioavailability ranges from 50 to about 80%. 80% is the highest for Cimzia. Their peak concentration is achieved 5 days, 2 days, 2 to 6 days, 2.3 days. So it takes a couple of days because of the slow subcutaneous absorption. For infliximab, the Tmax is obviously the end of the infusion. You can see that all, at least the ones that we have information on, have or follow linear PK. How can that be after I try to convince you that you have to worry about nonlinear PK for MAPs and for proteins in general? What that means is the doses are so high that they're exceeding the levels that you achieve, exceed by far the levels of TNF in the body. Okay, so the binding to the target really has become a small, the nonlinear, the sexual binding, has become a small part of the overall PK. So the overall PK across the board is linear, which obviously makes life easier. If you look at the volumes of distribution, you can see they range somewhere from 5 to maybe 10 liters. So they're all large proteins that have a tough time leaving the intravascular space, just like you would have expected. The big difference is in their clearance and their half-life. So you can see the what I would call the standard monoclonals, adalimumab and golimumab, the humanized ones. They have half-lives about 2 to 3 weeks and similar clearances. So that's basically what an endogenous IgG does. So their clearance is primarily via the FCRN mediated recycling. Let's look at the chimeric, okay, and you can see the chimeric one has a shorter half-life. So because of the fact that it has a murine component to it, it's eliminated more quickly, okay? On the other hand, the etanercept, the decoy receptor, has an even shorter half-life despite the fact that it's combined or conjugated with an Fc segment. Okay, so all those bioengineering, it helped it a little bit, but the half-life is still, of all 5 of them, it is the shortest. On the other hand, you can see that the pegylation of certolizumab gives it about a 2-week half-life, okay? Now, if we look at the various doses, you can see accordingly the adalimumab is given biweekly, subcutaneously. The infliximab is given loading dose, and then basically every 2 to 4 weeks, 2 to 8 weeks. The etanercept, the one with the shortest half-life, has to be given every week. Golimumab every 4 weeks, and certolizumab every 2 weeks. So the short half-life translates into the highest dose frequency for the etanercept. Okay, so the decoy receptor has to be given very frequently because it has the highest clearance and the shortest half-life despite the Fc conjugation. Now, if you look at the major drug-drug interactions, you can see this is not what you typically find for a small molecular weight drug where you look at other metabolic inhibitors, drug transport inhibitors. But here you basically have a pharmacokinetic, pharmacodynamic interaction. That methotrexate, which is typically given for the autoimmune diseases before you would start patients on those monoclonal antibodies, on those TNF antagonists. Methotrexate reduces the clearance for pretty much all of them with the exception of etanercept. And the mechanism behind that is that methotrexate reduces immune cells, and the immune cells are involved in the clearance via the Fc effector function. So by co-administering therapeutically methotrexate, you are reducing the clearance for at least 4 out of 5. Pharmacodynamically, as a result of co-administration of methotrexate being an immune suppressor, you would expect, and you can see the label states that the immune suppression is enhanced. Okay? So no major differences, as I said, with the possible exception of the fusion protein. Now the last thing to look at is the immunogenicity. So this is what's the incidence of either ADAs or infusion reactions, signs of allergic response. And I've highlighted here the 2 that stick out, because you can see, let's start with the ones that are basically very similar. Adalimumab, 5% ADA incidence, maybe lowering plasma concentrations and reduce efficacy. About the same incidence in certolizumab and golimumab. Again, the incidence is very similar. But then you look at the 2 problem children, if you like, infliximab and etanercept. Okay, so infliximab has a fairly high incidence of infusion reaction. That means during the infusion, people develop rashes, things like that, that may require treatment. Okay, that is a direct consequence of the fact that we're using a different species other than the humans as part of the molecule. And if you look at the fusion protein, that incidence is even higher. Okay, so you see those 2 are not only hampered by the fact that they have a fairly short half-life, they also, as a result of the fact that they're either artificial construct or that they contain chimeric, meaning murine sequences, they also have a higher incidence of immunogenicity. And I think that's it. I hope you not only enjoyed doing the exercise, but enjoyed listening to my lecture. I appreciate you paying attention to me, and if you've got any questions, as always, contact the program coordinator. Thank you.